Okay, so now I'm going to start with the DNA polymorphism. Okay, so what is a polymorphic DNA? Why is it necessary to understand that how the polymorphic DNA has been very much crucial for the identification of the evolutionary cell line or taxonomical significance in order to look into or examine into the cell line lineage starting from the old ancestors to the newer progenies or the successors, from the predecessors to the successors. And why is it necessary to understand in course of the DNA replication, the cell biology, mole biology, the entire central dogma, as well as the branches of proteomics and genomics, how it has played its important or vital role. Okay, so let's put the heading as DNA polymorphism. In case of DNA polymorphism, first let me draw or write a nucleotide sequence. Okay. Suppose I consider a 5' strand. This one let it be the 5' strand. Five prime to 3 prime, and this one be the 3' to 5'. This one let it be 3' to 5'. Now, in this is known as the leading strand. 5' to 3' is known as the sense strand, or the 3' to 5' is known as the antisense strand, the opposite of it. Now let me write a nucleotide sequence, a rough nucleotide sequence. Suppose GA, T, C, T, N, T, T, N, sorry, not N. Let me write all the nucleotides only. GATC, GTT, A, C, T again. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay. Suppose let this be a nucleotide sequence, a DNA nucleotide sequence, guanine, adenine, thymine, cytosine, and so on up to the thymine. Okay. Now the complementary of, we know that the antisense strand is always complementary, that it will have the opposite nucleotide or the complementary nucleotide, the base as per the base pairing rule or the base pairing fashion. So here the nucleotide sequence will be guanine will pair with cytosine, adenine will pair with thymine, thymine will pair with adenine, guanine, adenine, cytosine, adenine, adenine, thymine, guanine, guanine, again adenine. Okay. So this is what the complementary nucleotide sequence is. Now you see there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 base pairs almost present. This entire sequence of the nucleotide is about 11 base pair. 11 base pair. 11 base pairing means the 11 nitrogenous base pairing. Pairing means the pairing with the complementary nucleotide is present in this sequence. Now, this 11 base pair sequence in a DNA is often known as satellite body. Okay, these are known as satellite body. They are termed as satellite body because these bodies, this sequence of the DNAs have a remarkable function. They can repeat its sequence, repeat itself in that particular genome, or they can repeat itself in the genome of other cells in the body of that organism. That is, this sequence can present in different portions of the same chromosome, or the sequence can be present in the different portions of the chromosomes that are present within other cells within the body of that particular species or organism. Okay. And this why is this satellite body? Because satellite bodies, like in the chromosome, we have considered SAT bodies, sine acido thymonucleinico. Okay. This is a SAT body. The SAT bodies are very much essential in certain evolutionary function, there is evolutionary significance in the cell line lineage significance, because the SAT bodies are actually very much condensed portions of the chromatids as well as the chromonema, which has highly packaged conserved DNA structures or DNA strands that can help in producing certain kind of enzymes or certain kind of characteristic features of that particular chromosome or certain identification mark of that chromosome, which will be inherited from one generation to another, from from the parent generation or the mother generation to the daughter generation. So that satellite body is very much important. Now this is one form of SAT body, I mean the sequence of the SAT body. Generally these satellite bodies are known as mini satellites. Mini satellites. Mini satellites, these are almost 11 to 60 base pairs. It's 11 to 60 base pairs in the sequence. And within these mini satellites, there is also another type of satellite body, which is a subsection of this mini satellite body, and those are known as microsatellites. Microsatellites. These are about 1 to 6 base pair. 1 to 6 base pair. These are microsatellites, very small, only 6 base pair of nucleotides. In so many genes, so many in this entire 2 meter, 2.2 meter. Human genome is about chromosome is about 2.2 meter, right? So in the 2.2 meter and having so much of base pairs, like 6.6 into 10 to the power 9 or almost 10 to the power 10 base pairs are almost present there. So in this so many numbers of base pairs, just think 6.6 into 10 to the power 9, if we consider billions of base pairs, billions, trillions of base pairs are there. In this only 11 to 60, which is not even coming into any countable percentage or any handsome countable percentage, and the microsatellite 1 to 6 is almost not even something percent, 0.00 or 0.01% of the entire thing, if we consider the ratio. Okay, so the significance of these two satellite bodies are, like as I told, the SAT body. Satellite body is a conserved sequence of the chromosome, which is which is a marker or identification of the evolutionary significance inherited from one generation to another, because they help in producing certain characteristic feature of the chromosome as well as that organism, produce certain enzymes, help in storing certain kind of produced amino acids and all, produce proteins and all, hormones and all, or some other characteristic features or bodily functions and all. Those are being produced, kept conserved within that chromosome, within the satellite body. Now this microsatellite body, let us consider the base pair starting from this position. This is one base pair, this is another, this is third, fourth, fifth, sixth, seventh, 8, 9, 10, 11. Now we consider 6, okay. From here we consider up to here, this position. This is almost 6 base pair, which is called as the microsatellite. This is the microsatellite, and this one is the mini satellite. Now it is seen that these satellite bodies are repeated in the same fashion in other parts of the DNA molecule. Okay, other parts of the chromosome. That is, the DNA means the chromosome molecule only, whatever it is. So I write this entire structure again here also. I repeat this entire sequence here again here. It will be G, A, T, C, T, G, T, T, A, C, T, and I write the complementary sequence also, C, T, A, G, A, C, A, T, GA. Okay. Now, in this case, what happens is, in this entire repeated sequence of the DNA, in this entire repetitive sequence of the DNA, there would be, it is not always that this sequence would be the same exactly in all the repetitions within the same genome. It will be not be that this entire sequence will be repeated as it is. I mean, the base pairing will be as it is in all the repetitions within the same genome, within the same chromosome or the different chromosomes of different cells within the body of that particular species or organism. Okay, there would be a different change in the base pairing, change in the structure. Suppose here, in case of G, we can have, let me write, in case of G, here it is A, and the complementary nucleotide would be T. Okay. So you see that this nucleotide which is present in the middle has changed, but the starting nucleotide and the ending nucleotide sequence is same. This phenomena is called single nucleotide polymorphism. The single nucleotide polymorphism. Polymorphism means the structure of the DNA sequence would be different. The structure of the DNA sequence means the repetitive sequence. These sequences are known as repetitive DNA. Repetitive DNA sequence. Okay. The structure or the sequence of the repetitive DNA would be different would be different throughout the entire chromosome, where the sequence is repeating itself, or throughout the chromosomes or the genome present in other cells in the body of that same organism. Okay, this is that is actually what polymorphic DNA is, or polymorphism is. Now here single nucleotide polymorphism happen. What if I change this sequence also here. I write here guanine and cytosine. Here also I change the sequence. I write here thymine, and here I write adenine. Okay. In that case, you can see that this sequence, and this sequence, majorly the second and sixth, and the 10th base pairing, second position base pair, sixth position, second, sixth and 10th base pair, has changed. So the structure of the sequence has changed. The structure means here this A, this particular structure, this particular sequence, okay, this one and this one, this particular sequence of 3 nucleotides, here also 3 nucleotides, this nucleotide and this nucleotide is same here also and here also, and in this position here also. Okay. Only the nucleotide or the base pair at the second and the sixth and the 10th position have showed difference. This phenomena is known as multiple nucleotide polymorphism. Multiple nucleotide polymorphism. Okay, so this is what, now this sequence, this multiple nucleotide polymorphism, this is what the DNA polymorphism is, might be present in other sequences also. Like if I draw another sequence here, okay, and here also if I draw another sequence, this is 5 prime, 3 prime, 3 prime, 5 prime, 5 prime, 3 prime, 3 prime, 5 prime. This same sequence would have almost all the sequences similar. All the sequences similar it will have as it is. As it is, it will have all the sequences similar, but the variation like which happened here in the 2nd, 6th and 10th position might not happen in these nucleotides. Here, suppose the variation is happening at the first position the variation is happening, and at the 7th position is happening. And in this nucleotide the variation is happening in the 2nd position, and it is happening in the, here it is happening in the 9th position. But rest of the nucleotides, the majority of the nucleotides or the base pairs would be same. And only this particular structural body, which are known as the mini satellite or the microsatellite, will show the evolutionary significance that a certain type of base pairing has been repeated with some aberrations, with some chromosomal aberrations or change in its structural conformity or structural organization. It has been repeated since time immemorial from our ancestors. That's why that is producing a particular characteristic feature that is very much similar to our ancestors, and that is producing a particular enzyme or conservative structure of protein or producing a particular biomolecule that is very much important for our bodily function. It is producing a particular characteristic feature clearly that resembles that of our ancestors or the predecessors. So this is what this is what actually the polymorphic DNA is, and this polymorphism is almost present in the entire human biosphere. In almost all the human beings who are present on Earth, this polymorphic DNA is present in almost everybody. It is present, and it is also found that in other organisms also the appearance of this polymorphism is very much evident. Okay, so this is what the DNA polymorphism is all about, and how the mini satellite and microsatellite are repeating itself. These things are called simple sequence repeat. Simple sequence repeat. Okay, means SSR. SSR. Simple sequence repeat means these simple sequences are repeating itself. That's why they are not known as SSR. And there is another terminology called VNTR, variable number tandem repeat. Variable number tandem repeat, VNTR, which is majorly comprising of the mini satellite. Okay, these microsatellites are actually known as the simple sequence repeats. Often they are known, and these mini satellites are actually the variable number tandem repeats, because the number of base pair is more in this case. In case of the mini satellite, the number of base pair is more. Okay, this is what the terminologies are, and how the polymorphic DNA or the DNA polymorphism is happening within this repetitive conformation or the repetitive DNA. Okay, so this is all about how the repetitive DNA is showing its polymorphism is nothing but the repetitive DNA is only showing the polymorphism with different in its sequential structure. That is how the polymorphic DNA or the DNA polymorphism phenomena comes into being, right?